My name is Jensen Glass, and I'm going to be presenting my microbiology end-of-term project on the filoviruses. I'd like to start by introducing the classifications of the filoviruses. Filoviruses belong to the order Mononaega viralis, to the family Filoviridae, and to the genus Filovirus. Filoviruses have two known serotypes, the Marburg serotype and the Ebola serotype. Within the Ebola serotype, there are three distinct subtypes, the Zaire subtype, the Sudan subtype, and the Reston subtype, all named for the location with which they were identified in. Filoviruses are filamentous in structure, with branched, circular, U, and 6-shaped particles, which is unique to the viruses and is used for their classification. They are envelope viruses as well. As for a genome, the filoviruses contain a negative sense, single-stranded, linear RNA genome about 19 kilobytes in length. On the outside of the virion structure are glycoprotein spikes that are useful in the adhesion of the virus to host cells. The nucleocapsid contains the viral RNA and the viral structure of proteins, 5 of which to be exact, and 2 matrix proteins. 2 of the structural proteins are the nucleoprotein NP and the glycoprotein GP. Nucleoproteins are the major structural component of the ribonucleoprotein complex and is also known as the major structural phosphoprotein, which forms to encase the RNA genome. Glycoproteins form the spikes on the virion surface and assist in cell receptor and membrane binding. It is also known as the viral antigen. 3 other structural proteins are the L large protein, the VP35 protein, and the VP30 protein. The large L protein is known to be the RNA-dependent RNA polymerase of the virus and is involved in enzymatic functions of the virus as well. The VP30 protein is a minor nucleoprotein and is the functional unit in encapsulation and is also cofactor for the enzymes transcriptase and replicase, which are important in the processes of both replication and transcription. The VP35 protein functions as other P proteins and other RNA viruses would. The P proteins are known as polymerase proteins and assist in polymerase complex functions. 2 more proteins involved in the structure of the matrix of the virion is the VP40 protein and the VP24 protein. These 2 proteins are not associated with the RNP. The VP40 protein is a primary matrix protein, and the VP24 protein is a secondary matrix protein and helps to link the membrane proteins to the RNP. Here is a picture of the structure of the virion of the filoviruses. This is intended for visualization purposes. As you can see, all of the proteins that we just discussed are on this picture, showing you the location of the proteins on the structure of the virion. The filoviruses are known to have a zoonotic nature, which means that they are transmitted from an animal reservoir to a human host. A large problem of nosocomial transmission is found with these viruses, and contaminated medical and laboratory equipment are huge contributors to infection. When an outbreak occurs, they are known as endemic outbreaks due to the outbreaks occurring in only a specific region. All sources of the filoviruses known have African origin, and outbreaks always occur in African countries unless spread of the virus occurs. The reservoir of the filoviruses is currently unknown, but scientists believe bats, primates, ticks, and guinea pigs may be the carriers. The viruses infect host cells by adhering to the outside of the host cell by the use of the glycoprotein spikes on the virion surface. The virus particles then enter the cell through endocytosis. Once inside, the virus copies and transcribes its viral RNA in the cytoplasm of the host cell. Once the virus has finished replication and transcription, the virions are released from the cell through the process of budding. Filoviruses can cause major complications to non-human primates and human hosts. The release of cytokine from the immune response of the immune system and macrophages are unable to be controlled and cause both severe bleeding internally and inflammation, which leads to the coagulation cascade. Viral replication itself causes both bleeding and necrotization of the internal organs. As a result of the viral presence, the immune system cells are directly affected and are destroyed. The Ebola virus is one of the two famous diseases brought about by the filoviruses. The Ebola virus is linked directly to African origins and was named after where the organism was identified in 1976. Depending on the subtype of the virus, the mortality rate ranges from 30 to 90% according to the CDC, with the Zaire strains having the highest and the Reston strain having low pathogenicity in humans. Ebola is known to infect non-human primates and humans only. The Ebola virus is spread through contact with an infected human or animal, through aerosols and droplets, and through contaminated equipment. The Marburg virus, also caused by filoviruses and famous, was first identified in Marburg, Germany in 1967 during a laboratory experiment consisting of primates from Africa. The Marburg virus was the first of the filoviruses that was identified. According to the CDC, the mortality rate is 23 to 90%, which is slightly lower than that of the Ebola virus, but the reasoning is still unknown. The virus is spread through contact with infected animals and humans, aerosols, droplets, and from contamination from the remains of dead organisms. The symptoms of both infections have a rapid onset of about 8 to 10 days, according to survival. The symptoms include, but are not limited to, rash, fever, dizziness, severe body and headaches, and difficulty breathing. Later in the infection, symptoms such as shock can occur as a result of the excessive cytokine release from the immune system, comas, and bleeding from the eyes, mouth, and the nose. A major symptom of both viral infections and leading cause of severity is a hemorrhagic fever. Hemorrhagic fever interferes with the body's ability to clot blood and to stop bleeding. The HFs are the cause of internal bleeding in the organs, which leads to organ system failure. If severe enough and dependent on the strain of the virus, death can occur from these symptoms. Currently, and unfortunately, there are no known treatments and cures for both the Ebola and the Marburg viruses. Patients are treated by the treatment of symptoms as they occur to help avoid further complications and to help the patient be as comfortable as possible. There are also no known vaccines or preventative measures to protect and control the filovirus. Proper sterilization methods, use of HEPA filters, and the isolation of patients or infected individuals can help prevent viral transmission from researchers and medical facilities to the outside world. As I am presenting this PowerPoint, researchers all across the world are trying to find effective preventative measures, such as vaccines and treatment methods, for the virus itself rather than for the symptoms. Due to this research, more and more about the filoviruses are being learned and the knowledge is being applied. However, much is still unknown about the filoviruses due to the BSL-4 classification, making them extremely harmful and difficult to work with. The number of outbreaks is generally small and are usually contained within African countries, and also do not last very long. The cause of these outbreaks, however, are still unknown and happen rather spontaneously. There is also possible evidence to the emergence of varying subtypes of Ebola yet to be identified. While unknown to us now, the filoviruses may one day be less of a mystery and provide the necessary information to save lives and eradicate the diseases caused by the filoviruses. That concludes my presentation. Thank you for listening, and thank you for watching.